In part two of our video on speciation, we're really going to look at continue to look at what makes a species, as well as some of the different rates of how species can be made in terms of how quickly this can actually happen. And there's some different opinions and thoughts on that actual process. And so to start out with this video, let's define a species as a group of population whose members have the potential to interbreed in nature and produce viable fertile offspring, but do not produce viable fertile offspring with members of other such groups. And so the key portion of this here, again, is the ability to interbreed in nature and produce viable fertile offspring. If two organisms come together and they reproduce, but that offspring isn't fertile, meaning that it can't reproduce, it's kind of a dead end, essentially, kind of like a mule or a liger. Not being able to reproduce those hybrids. And so the important part is for it to be able to reproduce with another organism and to produce fertile offspring. Now, additionally, in nature, you think about dogs, going back to dogs, for example. If a really large dog, like maybe a big German Shepherd or Rottweiler, comes in contact with a small dog like a Chihuahua, the possibility of them breeding in nature is probably pretty slim. And so, this could eventually, years from now, go on to actually produce different species where they're not going to interbreed. But because of these different in physical characteristics and sizes, probably not going to see a lot of breeding between the two of them. And so the process of actually making a species, or the production of a species, is a term called speciation. And that is the process of forming a new species by splitting of an existing species. And we're going to look at two different ways that this can occur. The first is allopatric speciation and sympatric speciation. And in allopatric speciation, what we're seeing happening is gene flow is going to be interrupted within a population, and primarily due to some sort of geographic isolation. So let's look at an example. And this is an example that's been done in the lab before. If we take a group of fruit flies right here, and we split them into two different subpopulations and provide one maltose food and one starch food. So we're using two different food sources. And over many, many generations, if we allow these to survive and to reproduce, and after numerous generations, because of natural selection and genetic drift and potentially mutations arising in these different subpopulations, it's going to alter the allele frequencies. And this can lead to reproductive isolation. This is what was actually sought to occur. These two different subpopulations changed enough that there was reproductive isolation, so that when they were reintroduced with one another, the fruit flies that had been in the starch food or had been provided the starch food only continued to reproduce with those that also were found in the starch food, and vice versa for the maltose. And so a good definition of this is that allopatric speciation is the formation of new species while geographically isolated from parent populations. And ways that we see this actually occurring in nature, environmental changes causes maybe a geographic region to be split by a river, or the development of a mountain range, or a lake being divided into two separate ponds or something like that. An example of this in nature that we've actually seen is in a species of fish, mosquito fish found in the Bahamas. And if you look at these two different images here, the red and the blue, you'll notice that there are some differences in their physical characteristics of their tail regions right here. Right here, this one looks a little bit thicker, and this one's a little bit longer and not quite as thick. Well, what's happening? This mosquito fish population was divided into two different subpopulations. And in these two different subpopulations in different ponds, there were some different selection pressures. In this pond, where the mosquito fish now have a little bit larger tail section, there was an increased amount of predation. And so through natural selection and through multiple generations, these fish have changed because of those surviving. The population has changed so that fish have larger tail fin regions, which helps provide them fast bursts of speed to escape predators, which makes sense. In our other subpopulation, we notice that the fish have a little bit longer and not quite as a thick tail fin region. And what this does is it provides the fish a ability to swim longer and at more consistent rate and distances. And this is because there's less predation in the pond, in the area, their environment where they live. And so because of this division, geographic division between an original one single population creating two subpopulations, through multiple generations and different factors affecting these different subpopulations, we've seen actual physical differences between the two. The second form of speciation that we're going to look at is sympatric speciation. And this is essentially the same idea. It's the formation of a new species from a subset of parent species, but this is without geographic isolation. And this is definitely possible. Allopatric speciation is probably the more commonly thought of. You think of a population getting split into two. That's usually, you think of it as an example through geographic, but it's possible for this to occur without geographic isolation. And so let's use an example here. Let's say we have a little dinosaur population, and for some reason, some sort of sexual selection occurs where the little green dinosaurs fall in love with other little green dinosaurs, and little blue dinosaurs fall in love with other little blue dinosaurs, and they reproduce. And so the green color, the green allele types are reproducing with other green allele types, and vice versa for the blue. And so after a number of generations and after a number of time, we see this continue to happen. So we have two different subsets of the population, but they're in the same geographic region. They're not split by some sort of geographic or physical barrier. And this can also be caused by polyploidy, which usually happens in plants, but we'll talk about a little bit more in a minute. Temporal isolation, in which rather than there being some sort of geographic isolation separating individuals within the population, it's some sort of a timing in terms of when they're actually reproducing, in terms of time. Sexual selection, as we mentioned. A great example of this in nature is with different cichlid fish in Lake Victoria in Africa. There's a huge diversity and variety of different cichlid fish, the freshwater fish actually that I have in my classroom. And because of this huge diversity, or this diversity is caused primarily through sympatric speciation, all in the same region, but because of different selection, sexual selections or temporal isolations, we've seen a number of different species develop over generations. So polyploidy and speciation, another process that can produce speciation. And polyploidy is an organism that has more than two sets of chromosomes. So haploid would be those cells that would be gamete cells in humans. For example, two haploid sperm and egg come together to make a diploid full set. And so that's what we see in humans. And sometimes through mistakes in meiosis or in some other occurrences, we can see triploid or tetraploid, or sometimes even more. Strawberries, for example, have 8 octoploid. They have 8 sets of chromosomes, and this is usually occurring in plants. Sometimes in some asexually reproducing animals, and sometimes in humans through asexual reproduction. And so what we see happen is these additional sets of chromosomes can come from errors of meiosis, and they can create diploid gametes. And what this does is, for example, let's say that there is a plant species that through a mistake in meiosis becomes tetraploid, and so they have 4 sets of the chromosomes. They're not going to be able to reproduce with another plant that's not a tetraploid. Some plant species can reproduce asexually, so rather than reproducing sexually, this tetraploid plant now is going to reproduce asexually. This is a possibility of something that could happen. Eventually, you would think over time and just through chance, another of the same original plant species might also have that mistake in meiosis and become a tetraploid. And then those multiple tetraploids can reproduce sexually with one another. That is going to create and can create a different species. And so this is another way that we can see, would be another example of sympatric speciation, where we see within the same geographic region different species arise. An example is a type of potatoes. It has double the amount of chromosomes as the normal variation between these two different types of potato species. One of the last topics that we want to look at is something called adaptive radiation. And adaptive radiation is essentially the idea that there's an ancestor organism, and through time and through movement, geographic movement, and through different selection pressures, there's going to be different forms of species, different closely related, generally, species that develop from this ancestor species. And this is the idea that there's radiating from a central ancestor, and by doing so and by dispersing, these individuals have become adapted to their specific environment. So they're adapting to their specific environments and selection pressures, and thus become different species. Some similarities of homologous structures due to common ancestry is one way that we can see this actually happening. And we'll talk about homologous structures a little bit more with convergent and divergent evolution, which is looking at kind of our last main topic for this. Convergent evolution is independent evolution of similar features in different species due to similar environments. So that's going to be this one right here, B. And so we have two different species, and because of similar selection pressures within the environment, we see similar features develop. And a great example of this is the comparison of sugar gliders in Australia and flying squirrels in North America. You notice if you look at these two pictures, they have some very striking similarities in their ability to glide from tree branches to tree branches, but they're not related. They're not. They don't share a common ancestor. It's because of similar environmental selection pressures that they've developed these webbed appendages, basically between their appendages, in order to allow them to glide and to almost fly. That is convergent evolution. We have two moving towards each other with similar characteristics. The opposite of that is divergent evolution. That's going to be A right here. And divergent evolution is the development of different features in two or more different species who share a common ancestor. And so this is also going along with adaptive radiation. Because of a common ancestor, and through migration and spreading out geographically and different selection pressures, we see similar characteristics, but they're slightly different, maybe function. That's because they come from a similar ancestor. And so divergent evolution gives rise to homologous structures. To give you a couple of quick definitions for this, convergent evolution, independent evolution of similar features in different species due to similar environments. Divergent evolution is the gradual evolution of different features in two different species who share a common ancestor due to different environments. And a nice summary of that is right here, where we see convergent, divergent, and then parallel evolution. The last topic that I'd like to discuss is really looking at the rate, or how quickly we can see organisms change, and understanding some different thoughts behind this. And the first idea is something called gradualism, and this is really what Darwin was suggesting. And this is a slow, gradual change that occurs slowly over time. And so this would be organisms slowly changing over multiple generations, thousands, hundred thousands, millions of years, developing a different species. But sometimes the fossil record doesn't really match with that. And so in the 1980s, 1990s, another individual, Stephen Jay Gould, suggested this idea of punctuated equilibrium. And punctuated equilibrium is basically periods of no change followed by quick bursts of large amounts of change in terms of the number of species and diversity. And so there's periods of stasis where there's no change until there's events that disrupt that period of stasis, and it disrupts the equilibrium or balance. So there's sudden change in terms of geological time, and would still take obviously many generations to occur, possibly thousands of years, but in terms of overall the lifespan of Earth and life on Earth, it's relatively quickly. And so if we look at some phylogeny, a tree, to kind of demonstrate this punctuated equilibrium, you would see this by very abrupt and specific branching patterns, whereas gradualism, we're going to see a little bit slower and a little bit less distinct bands. So let's look at that as an example with butterfly colors here. If we're looking at gradualism, this would be the original parent species, and then slowly we see the development of these different colors. Whereas the opposite of that with punctuated equilibrium, we see this original ancestor species, and events occur that disrupt that equilibrium, and so we then see some very distinct different colors arise due to some sort of environmental or selection pressure. We're going to discuss this topic a little bit more in class, but this is kind of summarizing the main topic points for speciation, causes of speciation, and then rates, and how quickly this can occur.